What's up, distinguished boys and girls? Welcome back to my channel. I'm reading straight from my PDF. I'll link it in the comments, so make sure you go and check it. In this video, we will discuss the nucleus. So this is the structure of the nucleus that you can see here. We have inside of the nucleus, we have something called the nucleoplasm. You can think about the nucleoplasm being very similar to the cytoplasm. It's basically an aqueous solution or a fluid-like solution that suspend the nucleolus and the chromatin network, or the chromatin filaments. So if they ask you about the function, it basically suspends the nucleolus and the chromatin network, or you can describe it as bathe the you can describe the nucleolus and the chromatin network bathe in the nucleoplasm, just how like organelles bathe in the the cytoplasm. Do not confuse the nucleus with the nucleolus. They sound similar, but they are not the same. The nucleus is this spherical structure that you can see here, which contains the nucleoplasm and contains the nucleolus and contains the genetic material, which is represented by the chromatin network. The nucleolus is this part here that is responsible for the formation of the ribosomes, which in turn builds the proteins. The plural form of the nucleus is the nuclei, and the plural form of the nucleolus is the nucleoli. If they ask you about the function of the nucleolus, it just has a vital role in the formation of ribosomes, which in turn builds proteins. And if they ask you about the location of the nucleolus, it's inside of the nucleus, suspended in the nucleoplasm. I want to discuss the chromatin network and the chromatin fibers. What is the difference between the chromatin and chromosome? Now, the cell during its natural or its normal phase, it has the genetic material in the form of chromatin network. The chromatin network will condense to form the chromosomes only during cell division, usually at the metaphase. So, chromatin networks condense into chromosomes during cell division. That's why when we describe this here, we just describe it as chromatin, but not chromosomes. In order to fit the nucleus, it needs to be in the thin form of chromatin instead of thick chromosomes. One more thing is the nuclear envelope, or the nuclear membrane, which is just a membrane that surrounds the nucleus, and it contains pores. The nuclear pores. The nuclear pores are like gaps within the membrane that control the movement of material between the nucleus and the cytoplasm. So that's it. That's the basic things that we can talk about. Another very common question you can get is give an example for each of the following, and they would tell you a cell without a nucleus. It will be adult red blood cells. I want to clarify that red blood cells come from precursor cells that actually did have a nucleus at some point, but they lose this nucleus as they become the adult form or the mature form of the red blood cells. Because any structure without a nucleus, we cannot really consider it as a cell. So any cell without a nucleus come from a precursor cell that had a nucleus. This is also the reason why the life duration of the adult red blood cells is very short. It's only 120 days. They live only for 120 days. So if they tell you to justify or explain, the red blood cells have a very short life duration, it's because they lose their nucleus as they become adult. And the nucleus is the organelle that is responsible for keeping the cell alive. If they tell you, give an example for each of the following, and they will tell you a cell with irregular shape nucleus, it's white blood cells. Or they can tell you a variant nucleus shape, white blood cells. A cell with two nuclei, or dinuclear, as the book mentions it, it's the cartilage cells, liver cells, and muscle cells. A cell with centrally placed nucleus, meaning the nucleus pretty much placed in the center of the cell, it's stem cells. Stem cells are undifferentiated cells that can later differentiate into other types of cells. A cell with a site or peripheral location of the nucleus, you will just write down secretory cells, like fatty cells and mucus cells. Again, the questions that you can have here, either a blank question, being those blanks, or give an example for each of the following. I've also seen this here, a question, where they put the stem cells and they will just tell you to choose the correct answer between the brackets. And they put like the stem cells have a nucleus that placed, and they put in the bracket superficial, peripheral, or central. The answer is centrally placed nucleus. This is also found in the chapter questions, which I also provide in this PDF. The book describes the nucleus as the most important and the largest organelle in the cell. So also be careful of blank questions here, or give an example for each of the following question. The nucleus usually possesses a single nucleolus, but it's possible to have more than one nucleolus. For example, in the onion cell nucleus, we can have 4 nucleoli here. If they tell you to give an example for each of the following, and a cell without a nuclear envelope, we discussed this in the very beginning, and we said that prokaryotic cells, like bacteria and blue-green algae, do not have a nuclear envelope or a nuclear membrane. Instead, they just have a nuclear matter or a nuclear zone, or we call it the nucleoid, where the genetic material is just spread without a nucleus. If they ask you about the chemical composition of the nucleolus, it's RNA and proteins. If they ask you about the function of the nucleolus, it plays a vital role for ribosome formation, which is responsible for protein formation. If they ask you about the location of the ribosomes, you want to write down that it is found throughout the cytoplasm in both prokaryotic and eukaryotic cell, usually at the surface of the rough endoplasmic reticulum. In the exam, they want to see the word rough endoplasmic reticulum. They do not want to see they want to see the rough endoplasmic reticulum. This is the word the examiners will be looking for. So make sure you mention it. Again, nuclear envelope function. It regulates the exchange of material between the nucleus and the cytoplasm through the pores or micropores that allow the passage of particles. And this membrane has selective permeability. We will discuss permeability in this video. Now let's talk about the chromosome numbers. I want you to understand we have somatic cells and reproductive cells, or the gametes. Somatic cells are represented by the body cells, any cell from the body. Skin cells, liver cells, lung cells, blood cells, nerve cells, etc. But the reproductive or gamete cells are represented by the sperm cells in males and the egg cells in females. The somatic cells have a two set of chromosomes. We call it diploid. And the gametes have only one set of chromosomes, and we call it haploid. The number of chromosomes in somatic cells in humans is 46 chromosomes, 23 coming from the mother and 23 coming from the father. And the sum will equal to 46. But in the gametes, let's say in the sperm cell or the egg cell, the chromosome number is only 23. So 23 chromosome in the sperm cell and 23 chromosome in the egg cell. So when the sperm cell will meet the egg cell during reproduction, it will form something we call zygote. The zygote, okay? The zygote will ultimately have a two set of chromosomes, 23 from the sperm plus 23 from the egg, and will equals to 46. So it will eventually make a somatic cell to make a new individual. So if you want to extract the number of chromosomes of the somatic cell from the number of chromosomes from the reproductive cell, just multiply it by 2. And if you want to extract the number of chromosomes of the reproductive cells from the number of chromosomes of the somatic cells, you will just divide it by 2. The simplest example is the humans, 46. Just if they give you the reproductive cells having 23, just multiply it by 2, and it will equals to 46. Or the opposite, they can just give you 46 in the somatic, and they want the reproductive, just divide it by 2, and it will equals to 23. The books does not focuses on humans only. We also have other species that you have to memorize, because those numbers can show up in your exam as either a blank question or give an example for each of the following. For example, give an example of a somatic cell contains 2 chromosomes, and it will be the somatic cell of the Ascaris lumbricoides. We will just continue like this. So 2 chromosomes in somatic cells divided by 2. 2 divided by 2 equals 1, which equals to the number of chromosomes of the gametes. And the Spanish butterfly, 38 in somatic, 190 in gametes. Housefly, 20 in somatic. This is 12 in somatic and 6 in reproductive. The frog is 26 in somatic and 13 in gametes. For the pigeon, this is important as it showed up in one of the previous copies. They give an example for each of the following and a somatic cell that has 80 chromosomes? And the answer is just somatic cell of the pigeon. And of course, 80 divided by 2 is 40 in the gametes. The horse has 64 and 32. I love to think about it as the horse having exactly the numbers reversed of humans. So humans have 46 and horse have 64. So I think the horse is very easy to remember. Also note that the Ascaris lumbricoides having the least number of chromosomes among organisms, as they can ask you about the given example of an organism having the least set of chromosomes, or the least number of chromosomes. And the answer is just Ascaris lumbricoides. We can also have creative questions like they give you species of other animals that is not mentioned in the book, and they give you the, let's say, the somatic chromosome number, and they will tell you to extract the chromosome number in the gametes. You will just divide it by 2. Or the opposite scenario, where they give you the number of chromosomes in the gametes of one species, and they will tell you to extract the number of somatic cell. You will just multiply it by 2. Those questions are possible as they assess your comprehension. They make sure you understand the concept of the multiplication and division of the numbers. It's very easy, okay? There's nothing too special in those questions. Very easy to solve. I want to emphasize on the cell plate because I have, or I feel like it's going to show up in your exam. Again, the cell plate is only found in in the plant cells during cell division, originating from the protoplasm. And it grows from the center outward, unlike the animal cell, which is going to be from the outside to the inside, pinching the cell in order to divide it. And we call this the cleavage furrow. And here we have the contractile ring. The cell plate gives rise to the new cell wall of the new divided cell. And of course, to the middle lamella, which is also a part of the cell wall. So make sure you know this very, very well. So, let's start discussing the diffusion. The diffusion is the movement of ions or particles from high concentration regions to low concentration regions. For example, let's say that I have a membrane here, and let's say that I have ions here. Let's say that this is sodium ions, or whatever ion there is. Let's say that I have a lot of concentration here, high concentration in this region. And here I just have little concentration. The movement of those ions will typically be from the regions of high concentration like this, okay? So the movement will be like this. From the high concentration to the low concentration. That's the basic thing you need to know about the diffusion. Usually questions about diffusion and osmosis really depends on your understanding of the topic, because we can have very hard questions here. The examples of molecules that can spread through a cell membrane are gases, like the oxygen and carbon dioxide, and other materials that are soluble in fats, like the hydrocarbons and alcohols. Hydrocarbons mean just molecules that are made up of carbon atoms attached to hydrogen atoms. Basically, the basic structure of organic molecules. If they tell you to explain the experiment where we can observe the diffusion with the naked eyes, you'll just write down this. So basically, we have a water can and we fill it with water, and we put either copper sulfite or potassium permanganate, which is pigmented. It has a dye. It will typically diffuse throughout the water until it diffused throughout the whole water. But you need to pay attention that they spread on a short distance, and it hardly spread for a long distance, because distance of spreading particles is directly proportional to the square root of time for spread. So this is basically just telling you that the diffusion process is just going to depend on time. It is dependent on time. It will take time in order for those particles to diffuse throughout the solution. As we go, as time passes, we will reach the equilibrium state where the dye is actually distributed throughout the whole water. But it just needs time. It is dependent on time. This is just the fixed second law of diffusion, where it states that the distance a particle spreads, states that the distance a particle spreads is proportional to the square root of time. Here, as you can see it here. You do not need to know the name of the law or the formula. I'm just giving it to you to understand what is going on, because this is not mentioned in the book, so it's just extra thing that you do not need to memorize. Also, whenever you have an experiment question, always, and I mean always, provide or include drawings in with your answer. This is not just limited to biology, it's also applicable to physics, for example, in the activity question, and chemistry as well. Possible question is that they just tell you to draw something to illustrate the diffusion process, and you're just going to draw this. Permeability. No, permeability is the exchange of materials between the cell and its environment. We can classify the membranes into four types according to their permeability. We have permeable membrane, which just means that the membrane allows the passage of any material, regardless of their size or structure. The example mentioned is the cell wall. Of course, you need to pay attention to the blank question, as this here can be a blank question, or the example of the cell wall being a blank question. Also, the question that states what is, or give an example for each of the following, and they will tell you a permeable membrane. It's just the cell wall. The semi-permeable membrane and the selectively permeable membrane are very similar, and you can easily get them confused. Semi-permeable membrane means that they allow the passage of solvents more than the solutes. You need to understand the concept of solvent and solute. Solvent is the liquid material that usually the solutes dissolve in. The simplest example is water and sugar, or water and salt. So if we have water and sugar, the semi-permeable membrane will allow the passage of water more than sugar. And the selectively permeable membrane they selectively, from the name, they selectively allow the passage of certain materials according to the size of the particles. For example, if I have very large particles, very large protein particles, those large protein particles are not allowed to pass through the membrane here. And finally, we can have the non-permeable membrane, which do not allow the substance to pass through it. For example, the nylon. If they want to know the permeability of the cell membrane, I've seen some overlap in sources some of them describing it as a semi-permeable, and other sources describe it as selectively permeable. And the book itself describes the cell membrane as a semi-permeable in the osmosis definition, but they also put a previous question where they were asking about the permeability of the cell membrane, and the answer was selectively permeable. Now, what I can tell you is that the book probably considers the cell membrane as either selectively permeable or semi-permeable membrane, meaning they can consider both answers as correct for the plasma membrane. But personally, what I would choose, I would say that selectively permeable. The cell membrane is selectively permeable. It's more correct than just saying semi-permeable, in my opinion. But I would say both answers should be correct, as the book literally describes it as both. Another acceptable answer for the semi-permeable membrane is the cellophane. You can have comparison questions between the permeability of membranes. It's very easy. It's just 1 point. You can have any two membranes and just to compare them. So let's discuss osmosis. Osmosis is very similar to diffusion, as it is actually a case of diffusion. Now, osmosis cares about the movements of water through the semi-permeable membrane. Here, as you can see, the book mentions plasma membrane being a semi-permeable. So the movement of water is going to be according to the variance in concentration. So what does that mean? If I have, this is a membrane, and let's say that I have proteins here. Let's say I have a lot of proteins, okay? I have a lot of proteins particles here. And let's say that I have only one protein particle here. The movement of water will typically be like this. Okay, so the movements of water will typically be this way. So water will move from the area of high concentration of water to the area of low concentration of water. But the solutes here will be higher than the solutes in this site. Okay? But when we described the diffusion, we described it as the movement of particles from high concentration to low concentration. But if you remember that we said that here I gave an example specifically for proteins, because proteins, large particles of proteins, are not diffusible through a membrane. Okay? So that's why the water movements will take place instead of the solute movement. Okay? So if they tell you to explain an experiment that demonstrates osmosis, we will have this. So we have a water basin here and a water funnel here. And both are separated by a semi-permeable membrane. We use cellophane in this experiment. We will fill both the funnel and the basin with water. That water will be equal in both the basin and the funnel, in equal level. And then if we add sugar to the funnel, that will cause the concentration of solutes in the funnel higher than the concentration of solutes in the basin. So water will typically move to the funnel in order to equalize the water content in both the basin and the funnel. That's it. That's everything about it. Again, do not forget the drawings whenever you have an experiment question. We have something called the osmotic pressure and the other thing called the hydrostatic pressure. Now, the hydrostatic pressure is the pressure exerted by a fluid in the fluid within the fluid itself. For example, if I fill in this can with water, due to the force of gravity, I will have the pressure higher as I go deeper within the can. So the pressure, as I go deep within the can, the pressure will be higher. This is just due to the force of gravity. And if I put holes, and those we call jets, this jet is going to be strongest because here I have the highest pressure. So water will typically flow in this jet. This jet will be the weakest as my pressure here is very weak. So we will have only little fluid that will flow through this jet. But this jet is gonna be very strong. As I go deeper, I'm gonna have higher pressure. And this pressure we call hydrostatic pressure. It's just due to the force of gravity. But the osmotic pressure is different. Here I actually meant that the osmotic gradient is the thing we need to pull in water, but the osmotic pressure is the thing we need to oppose this water movement. So we don't actually need the osmotic pressure to pull in the fluid. It's the other way around. The osmotic pressure as the minimum pressure that we need to be applied to a solution to prevent the entry of water through a semi-permeable membrane. For example, I need a force here, another force we call the osmotic pressure, in order to stop the water movement through this semi-permeable membrane. This is what we call osmotic pressure. So osmotic pressure is the pressure that will oppose the osmosis, that will prevent the water from diffusing through a membrane. When hydrostatic pressure equals to the osmotic pressure, the movement of water will stop. There will not be net movement. Of course, we have the solutions are classified into three according to the osmotic concentration: isotonic, hypotonic, and hypertonic. Okay, so in the isotonic solution, we will have the concentration of water outside and inside of the cell is equal. So there will not be a net movement of water. And the cell will maintain its shape. But if I have a hypertonic solution, the solution will have a lower concentration of solutes outside of the cell compared to the inside of the cytoplasm. Again, I have low concentration outside of the cell, and the high concentration of solutes is inside of the cell. So here, the typical movement, as I told you, will be into the inside of the cell. Water movement will move inside of the cell as I have higher concentration of solutes, non-permeable solutes like proteins. This will cause the cell to swell and eventually rupture, as you can see here. And finally, we will have the hypertonic solution, where the solute concentration is high outside of the cell compared to the inside. So I have my concentration outside is high compared to the inside. This will cause the water movement from inside to the outside. This will cause the cell to shrink, as you can see here. And in plasma cells, in plant cells, as we have a cell wall, when the cells shrink, the cell membrane will detach from the cell wall. This process is called plasmolysis. You can have it as a definition, very important. But when water is reintroduced, this process will reverse, and we will just call it deplasmolysis. Finally, we will just describe the this is not finally, actually. Now we will just describe the active transport. Sometimes when we have the concentration inside of the cell, okay, the concentration inside of the cell of a solute is very high compared to the outside. So the typical movement here that you can imagine as we had in diffusion, that the solutes will diffuse from inside to the outside. But here, we can have the opposite. We can still have the solute movement from the outside, which is low concentration, to the inside, which has high concentration of solute. Even though this is opposite to the concept of diffusion, this is the opposite to the natural diffusion laws. This is because we have carriers in the cell membrane. So the carriers in the cell membrane will perform this process. Just know that the carriers in the active transport requires energy, unlike diffusion, which happens with no need of energy. But here, as we are going against the natural laws, we of course need energy to perform such a thing. So we will need ATP as a source of energy. Again, the location and function of carriers is important. The location is in the cell membrane of cells that perform active transport, and the function is just carry or transport particles or ions into the cell from the outside through a process known as active transport. We'll discuss phagocytosis real quick. I don't want to make this long. The phagocytosis is basically when the cell membrane performs a pouch-like structure within the membrane to surround a food particle. And then this will detach from the surface to the inside of the cytoplasm as a vesicle or vacuole. And then later, this vacuole will, or the food particles within the vacuole will be digested by the help of the lysosome enzymes. This phagocytosis also happens in the white blood cells during infections as they try to digest and eat the pathogens like bacteria. The pinocytosis is very similar to phagocytosis. It's the same concept, the same mechanism happening, but instead of a food particle, instead of a solid food particle, we will just have a liquid material. And finally, the exocytosis is the opposite to phagocytosis and pinocytosis. Because exocytosis, the vacuole or the vesicle will move from the inside to the outside. This happens when when we have undigested food particles and we just want it to get it out. Or this can also be the mechanism of hormones to secrete some, of glands to secrete some hormones. Okay, so this is also a mechanism of gland cells to secrete some hormones. It's by exocytosis. That's it for now. If you like the video, please subscribe to my channel. Share it with your friends, and thanks for watching.